Okay, good morning, class. So today in zoology lecture, we are going to see the topic about microbiology. So, as we studying all about the microorganism in our past lectures, and we see the how the subject microbiology come in existence with the study of microorganisms. We already study about the different aspect of the microorganism. We studied about how we can classify them in some manner, and basically classification we studied in field of the microbiology, or you can say in the purpose of different field of the microbiology. We studied about a diverse nature of microbiology. Also, we see how the history of microbiology happened in past 3 to 4 centuries. Also, how microbiological application are very diverse and very useful from the field of ecological field to the industrial field to medical field. So now here we have to see what is microbial phylogeny. So as the name suggesting here, phylogeny. So this is going to be dealing with the relationship among the microorganisms. Okay, each and everything in our whole environment, in our whole ecology, everything is related to one another. In any manner, they are related. Some they are having the same kind of structure. Some have some internal structure similarity. Some have some gene similarity. So, or sometimes they just have to depend on each other for their survival. So, in this manner, we needed this environment to survive together. So that's why this whole phylogenetic concept is based on the classification of the species in superior taxa and the construction of phylogenetic tree based on the evolution relationships. So now our aim and objective in this lecture that we have to study how we can make a phylogenetic tree that is based on the evolutionary relationship among the taxa. And this study is basically known as a phylogeny. Let's see the introduction and overview part. This phylogeny, this is a Greek word. You know that most of the words in the science, basically they can be derived from Latin word, Greek word, and some are very different from the origin. But most of the diagnosis of the naming that has been done by the Greek and Latin language. They are very ancient and developed kind of scientific language actually. So that's why the most of the term which we study here, we study the name of the Greek and Latin words. So the term phylogeny, that is a Greek word, and phylon, that possessing the meaning of the tribes or races or group. In a very appropriate, if we talking the phylon as a group, okay, group of a similar characteristic feature species, or you can say the species which are present inside the group possessing similar kind of characters. So basically, what we have to see the phylogeny. First, the group, and second term here, genesis. So genesis is always related for the generation something and origin something. So here also we are originating a species, and on the basis of their origin or on the basis of their development, we are going to put them in a specific kind of the group which are having the same similarities as this new species we have here. So basically, this refer to the evolutionary development of the species. Now, the phylum, the group, and their origin, that is based on the evolutionary developments, and it is based on the direct comparison of genetic material and gene products. So, if we sometime we can evaluate something just on the basis of their outer structure, how they are looking, but that is never sufficient. Even in the practical manner, in the social life, and also in the phase of the these microorganisms or plant or animals, we cannot just classify anything or we cannot just analyze something just by seeing them because they can falsify us. Okay, they can just give us some concept which we are going to fail on future. So that's why for our verification, for the validation, for getting the accurate thing, we need some digging, or you can say we have to go deeper. And by meaning the deeper, we have to analyze all the parameters for which we can analyze and find the more information we can got from these things. So finding the more information and getting deeper digging in the species, this is solely indicating towards the genetic material studies, because genetics is the thing which cannot tell a lie, or which cannot be falsified, or by which we whatever we are getting, and if we are getting it from the genetics, so keep it in mind that that thing will be 100% sure, because genetics have the accuracy. Morphology don't have that much accuracy as comparison to if we see the other parameters. Morphology don't have that luxury to provide the efficient thing, but genetics have. So that's why this whole phenomena behind phylogeny, or whole concept behind the phylogeny, why we are studying it. The main focus of our study is that we are have to, and we got the information which is reliable, and you can trust that information and you can proceed with that. Now, it is a bringing order to the diverse variety of organism present in the nature. So basically, there are a lot of organisms which are present around us. Okay, there is not a defined number. You cannot say like we are having this hundreds and thousands and lakhs. There is a number you cannot even count. Sometimes most of the species still now have been identified, but some of them still not. But even if they have been identified, we don't know yet in which kind of group we can place them. For so in bringing the order to the diverse variety of organisms present in nature, we have to classify them, because there are a lot of the species present around us. Okay, if we wanted to study each and every species, that cannot be possible. Like in your course of the bachelor in the science, or bachelor's in the pharma, or the courses that are related to the science, and if you wanted to make a drug, and if you wanted to see that how it is going to work on what kind of a specific species, so you cannot just going to test on each and every species. You just to take a model thing, or knowing that model thing, you know the specific character of that particular group in which those species align. So that's why we needed to, you can say, we needed to difference out. We needed to make them specific to a specific group. Then only we do not have to go through or go behind every each and every step. If we wanted to take a knowledge, we just go by that group, reading out their whole characterizing features, and after having the knowledge of their characterizing features, we will be able to know what kind of species are present in them. So in this manner, this will actually make easy for us for studying and determining the research or anything what we wanted to go in detail. That will be easy if we are going through the classification pattern. First one is based on the morphological characteristic. So, as this classification that is not new. Even in my last one lecture, I think in the plant taxonomy, I told you about how these classification parameters work. So basically, these classification parameters, which is present inside the classification, they are very ancient and very taxonomist, and you can say botanist if they are identifying the plant species, or even the microbiologist if they are identifying the microbiology. So they have been working on their classifications. So the initiation of these classification patterns, that has been done by the very first step I see morphology. That is a, you can say this is very common. When we even see something, the first thing we see their outer look. So the same thing the scientists have been done. When the idea of classification came into their mind, the very first thing what was in their mind is by looking the species. Just if they are finding some species is very tall, they are going to put them in a different group. If they think this is very short, they can put them into a different thing. On the basis of color, they are going to put them aside. Or on the basis of just morphological character. Or in the morphological characters, we are just talking about like the shape, size, and the color mostly, or the surface texture. So these are the things which come under the morphological analysis. So these morphological analysis was the very first kind of parameter which have been used for the classification. But that was not enough because just by seeing them you cannot tell everything about it. So that's why the second evolutionary relationship come under the process, that is phylogenetic classification. This is the key of all the problems you can say, or the loss even instead. So the phonetic classification, if we're talking about the like the accurate meaning of these terms, phonetic classification. You go through with the genetics, and we know the internal component of the genotype. We call them the genetics. But if we are talking about the outer appearance, we call them the phenotype, and we see them by the outer surface. So just we can call them the phenotype. So it is based on the mutual similarities of their phenotypic character. So basically, when we are talking about phenotypic classification, then we are just going to see that there have to be a different manner. So the phylogenetic classification. So these systems are based on the evolutionary relationship rather than the external appearance. So as we are talking about the phylogenetic, they are not just depend on the external features. They are rely on the evolutionary relationships. Evolutionary relationship, if we talk about what is evolution. So evolution is something that depend on a history, histological background, how their ancestor was and how in the present terms they are working. So if something is related to the ancient time and we are studying it now, within comparison to their ancestral origin, so that is a kind of evidence-based thing classification. So that's why this phylogenetic classification, this just not work on the similarity between the species inside a taxa, but also they work on the evolutionary pattern that has to be done for, you can say, if we wanted to do a efficient kind of the classification, these are the manners we have to follow. Next, we will see the phylogenetic tree. So, in the phylogenetic tree, these relationships are illustrated in the form of the branch diagram or the tree. So, as the tree look like, the same the phylogenetic tree we are talking about. But here, the tree is giving us the information. Okay, this is a source of information, and what kind of information they are going to provide us? They are going to provide us the information about the ancestral belonging and the similarity between the species in a group. So here, phylogenetic relationships are illustrated in the form of branch diagram, and a phylogenetic tree is a graph made of branches that meet of the branches and the roots. So this whole phylogenetic tree concept, that is alike of a dendrogram and histogram, if you heard about them. So these are the graphs that have consisting the branches and the roots. So each of everything in this phylogenetic tree, they have their own meaning, like the branches and the nodes. They have their own pattern, their own meaning. So the nodes we present here, the taxonomic units such as species, genes, or external nodes, they are present and the means they are present at the end of the branches. They will be going to represent the living organisms. So here, the node represented the taxonomic unit. Taxonomic unit. What is a taxonomic unit? So basically, taxonomic unit is like you heard about the kingdom, phylum, divisions, order, genus, species. These are the taxonomic units. Okay, and as the external node, if we are talking about here, which present at the end of the branch, they are going to represent the living organism which come under that specific division. So this is the major phenomena behind the phylogenetic tree, that they are going to give us a specific kind of taxonomic unit along with the living organism that has to be present in them. So the tree may have a time scale, or the length of the branches may represent the number of molecular change that have been taken place between the two nodes. So basically, this a kind of graph, and graph give us an analytical approach. So same here, this is not just a normal tree; they have their analytical aspects too. So how they can give us the analytical aspect? How can they explain us like what is their meaning? They are not just presenting some units; they are even giving us the data, and what kind of data we can got from a phylogenetic tree. So basically, they can function as a time scale. So the in when we're talking about a time scale or the length of the branches, so like there is a length of the branches like 1 centimeter, and for the 1 centimeter length, there will be you can say there will be a time period of like 100 years. Okay, so if we are having a branch that have a length of the 3 centimeter, we easily can calculate that there will be the 300 years old something. That if this phylogenetic tree are showing some ancestral relationship with their ancestor to the present time, so you can easily go through with their ancestral one. The number of the molecular changes also, not only the time they have been traveled, but also the molecular changes that has been occurred at that particular or in that time. This is evolution. Evolution is a series of mutation over a time period. So in this case, what we are having, this branch length, they're also telling us about what kind of molecular change has been seen in this particular time of era. So that's why this whole scenario of having nodes, branches, their length, giving us the time scale, and also telling us about the molecular changes that has been they have been through with. Even so, all these combining all these manners, we are having the tree which can be in two forms. It can be unrooted and it can be rooted. So now let's look what is the meaning of having an unrooted and rooted plant. So an unrooted tree that simply represent phylogenetic relationships, but it does not provide an evolutionary path. Unrooted, if we are having a phylogenetic tree, and this is the kind of unrooted. So, what their major perspective is to here, they are going us to provide the phylogenetic relationship. Means that they can going to tell us about the similarity between the taxa or similarity between the species, but they are never going to tell us how their ancestral origin takes place or from what they come from. What is their point of origin? What is their neogenesis? Okay, that is a major issue in the unrooted tree, that they never going to tell about their origin or their originogenesis. So that's why this unrooted tree, they can give us a one data point, but they are not as sufficient as the rooted phylogenetic tree. So there's a diagram here. You can see the diagram. This A diagram. Okay, so this is a diagram here. This is showing us about the a plant, but they are not showing us about any ancestral common ancestral gene. So they are having, by seeing this A, we can tell us about like this is a point and this is a B point, and they are having some similarities in between them. But we never going to find about their ancestral relationship. In the B, this is a rooted kind of phylogenetic tree. So in this rooted kind of phylogenetic tree, we can easily able to analyze that they are having this common ancestor behind them. So that's why they are more important, because they are not just giving a one point data to us. They are giving both the aspect of the data. They are giving us about the phylogenetic relationship, as well as they are giving us about the ancestral origin of these species. So simply specify the common ancestor for the four species or the direction of change. In contrast, the rooted tree does give a node that serves as a common ancestor and shows the development of four species from it. So, when we talk about the rooted plant, they give us the whole scenario about the ancestral being, but the A thing, unrooted, they will never going to give us the ancestral compensations. Now, a new term come in fashion here that is called domain. So what is domain? Actually, domain is a word that is not very old. This come is in last century, 20th century. This come in presence. Classification is old, but domain is new. In the domain, this is also a variant of the classification. So basically, how it come in picture. Advances in genomic DNA sequencing of the microorganism, biologists are increasingly adapting the classification of living organisms that recognize three domains. Now, what happened? Initially, we are classifying things on the basis of their major two or three parameters. Not very much parameter has been considered for the classification. Like if you remember about the plant taxonomy, all about the artificial classification given by Hugh Ferris and corrected by the Lianis. Basically, they have been working on some very limited parameters, but they have an idea that's why that they were important, but their classification was restricted to certain parameters. But now when we do all the research, do all the study, got all the parameters on which we can classify anything. So after having all these parameters, the most important parameter what we got, that is the genomics, as I already explained why genes are so strong. So that's why, because of these genes or differences inside their genetic makeup, or on the basis of their genetic makeup, we classify these three domains. So these three domains are taxonomic level higher than kingdom. And these domains in a taxonomic level, like a taxonomic level, what we put like the first kingdom, phylum, division, class, order, family, and genus and species. In this whole taxonomic level, we put this domain level higher than the kingdom, even in the as comparison to the higher to the kingdom. Archibacts are in one domain. The first domain is all about the archibacteria. EU bacteria in a second domain, okay, second EU bacteria in second domain, and eukaryotes in the third domain. So here we have these three main domains on the basis of our genetic makeup. So these domains are the archibacteria, EU bacteria, and third one is eukaryotes. So eukaryotes are the domain in which we are present. Or you can also say that eukaryotes are the domain which have a high diversity, or every kind of the very developed kind of the species present inside this domain. So this domain of the eukaryotes, they are going to further subdivided into the four kingdoms. organisms, plantae, animal, fungi, and protista. Other these two domains, they are not going to further divide into any kind of kingdom. They are divided into several classes, but they are not going to divide further in the kingdom, because kingdom is also not a small unit. Kingdom is a pretty large unit. Even you can see the whole plantae and whole kingdom of the animals. They are going to surround like the lakhs of the species inside them. So that's why they are not as small as it is looking here. This is the three domains, or you can say the three domain based on rRNA sequence analysis. So, on the basis of the rRNA sequence basis, we analyze them or we classify them in the three domains. So, these three domains, bacteria in the blue, Archaebacteria, archaea you can say, that is the pink, magenta pink, and the eukaryotes, they are in the yellow orange shade. So these are the things which we can study here, and the eukaryotes, you can see that we all come major things, or you can see the major developed organism, they all come under the eukaryotes. But as is in the zoology microbiology subject, we can assure that the two domains are completely devoted to the microorganisms, and these are the eubags and the archaea. These are the things which are completely devoted to the subject microbiology and the stream of microorganisms. Now we will be seeing the domains one by one. Archaebacteria, the very first here. The term Archaebacteria, also a Greek one. Archaeo means ancient and refer to the ancient origin of the group of bats. So basically, in this whole Archaebacteria thing, so in this Archaebacteria, we are having the bags which have a very ancient origin. Okay, so which seems to have diverse very early from the eubacteria, and you can see basically they are the ancestor of the eubacteria. The second domain, bacteria, they are the ancestor of them, or you can say by having some modification in the Archaebacteria, we got the eubacteria. So they are inhabited mostly in extreme environments. So why we are calling them the ancient one? Because the old is gold type thing. So old thing have more power for the sustain, or you can say more potential to sustain, or just live in a harsh condition. So basically, these archaebacteria, they're also pretty related to the very extreme kind of environment. Like what kind of extreme environment they can live into? They can be live into the methane production environment. Methanogens. Methanogens are the one which can have methane inside. Methane is a toxic. So simply if we going to in exposure of methane, we just going to die, or maybe just going to damage some of our organs. Extremophiles, they can be live in any kind of very extreme conditions. So extreme condition is like very high temperature. Extreme condition in the sense of very high salinity, or sometimes may very hot springs and very cold regions, or sometimes the pH level is very high. So anything which is not normal actually. So anything which is not normal, they can be lived in that condition. That's why we call them the extremophiles. And the third one, which are basically pretty normal, non-extreme archaebacteria. The other one which do not live in the extreme condition, but they are having the rRNA that is related to the archaebacteria. We put them also in this domain. So these are the major three categories of the archaebacteria. Now we were going to see the domain second, that is about bats or bacteria. They are also, you can say, except the bacteria which come under the archaebacteria, all bacteria come into the domain bats, eubacteria basically. So the eubacteria, they are the most abundant organism on earth. Okay, most abundant means they present everywhere, even our tongue, on the surface, on air, on the water, and the soil. Everywhere these insects are present. and we need them also. Not they are present just likely, they do not have to do anything. They have a very potential role to play. Okay, they have to play a major role in the carbon and sulfur cycling. Much of the world's photosynthesis is carried out by the UVACs, and however, certain groups of the UVACs are also responsible for many forms of disease. So as they're good for us, also they are bad for us. First, 90% of the bacteria are good for us. Only 10% of the bacteria which are having some pathogen capacity, or you can say pathogenicity, they can make us feel bad or create the diseases, so or become lethal. So that can be a character of the bacteria. Everything happens for a reason. So that's why if they are having 90% good to us, also the 10% which can harm us. Next year, the domain eukarya. Okay, so what happens inside the domain eukarya? It consists of four kingdoms. This is the one in which we belong. Eukarya is the most developed. So the first of which protista mostly unicellular like amoeba. So if we see the very first kingdom inside the eukaryotes that are protista, and mostly protisters are unicellular, very tiny appearance and look like the amoeba. But the three other domains, fungi, plantae, and animalia, they are having a wide range of the living being. They have the complex organism, they have the multicellular organism, they have the very high advanced feature inside the eukaryotes. So you can easily say that the potential diversity of organogenesis you can see in the domain eukaryotes. But this is not our whole topic to discuss about the domain eukarya. We just have to take overview and we already taken. So next step here, this is a diagrammatic representation. Like you can see, only one fourth of the part is called as bacteria and archaea, and most of the other part is called as a eukaryote. Now the phylogenetic relationship determining techniques. Till now, we studied what is phylogenetic. Now the next question is here that how we can study them. Like for studying everything in science, we need a technique to do it. Like if we wanted to see amplification, we use PCR. If we wanted to separate something out, we use centrifuge. If we wanted to separation, also we used chromatography. If we wanted to analyze something under at larger scale, we use microscope. So basically, there are several techniques which we needed to perform this study. So in here, for the phylogenetic relationship, these are the some parameters which we are going to study. Parsimony analysis. So what is parsimony analysis? Basically, parsimony analysis can work on a very minor scale. Like every species, if we compare them with another species, they have some variations in them. So basically, the miniature minimum sequence of changes or minimum amount of differences in between them, which can clear the differences between two species, that is called parsimony analysis. Like a person come to you and tell you that these two species are identical or these two species are same, but you are not agree with her. You tell no, it's not true. They are different, and they are different in many ways. But he just asks you that tell me some major important differences between these two. So now you are going to analyze these two and tell him the major important differences on which you can evidently prove your point by telling them that they are different species and these are their major differences. So, on the basis of very small sequence analysis, you can be able to find that these things are different. So that's why that kind of analysis can be called as parsimony analysis. So this is all about the parsimony analysis. Now let's go for the molecular chronometers. They are a little wider, but chronometer. Why we need a chronometer? This concept of having chronometer in our lives, this concept first suggested by Sunkrandel and Pauling in 1965. which is they are studying about the phylogeny and got with a technique with molecular chronometer. This is based on thought that the sequence of many rRNA and protein gradually change over time without destroying or severely altering their functions. By the way, this thought is very true and this happens. Majorly over the time period, like if we see a series of changes in our body, rRNA is present, ribosomal RNA, that is called rRNA. So they needed to change, or you can say they change over a time period every time. But in this manner of their changing, they do not delete anything. There is a term that is called modification. Like, okay, and sometimes we can say over having something. In that scenario, the simplest meaning is that when they are changing something, this change is not by deletion. Okay, they are not going to delete any sequence inside them, but they are just going to add some phenotypic effects, or you can say they are just going altering the effect of particular sequences. Sequence is same, but effects are different, and that happens over a time period. So this kind of concept that was given by Pauling and Zuckerberg, this for studying their, you can say for studying their alteration in the functional study in their changes, and basically these changes are linear with time. Like one change come in 100 years, second change come in 200 years, third change come in 300 years. So basically these are linear with the time. So linearity of these changes in accordance with the time, if we are going to analyze that thing, the molecular chronometer are the thing which give us the whole information of what kind of changes has been occurred inside the rRNA and proteins. So this whole thing or whole practice of how we can study the changes we can go through with the molecular chronometers. The third thing is their oligonucleotide signatures sequences. I tell you, they are so important. Oligonucleotide signature sequences actually they are very specific as a signature. Every person they have their own signature until we copy them. But still, every person have their very best signature sequences not inside our body. Also, if we do sign, that is very unique to person. Okay, so that's where the same oligonucleotide signature sequences. The same thing happened that this 16SR RNA unit. Okay, this 16SR RNA. They are very specific to each and every species. Like if we are having a group of 10 people, and if we are going to analyze their 16 srRNA, they will be very specific like the DNA. But what happened when we study the DNA? That is a complex process, but nowadays this is an easy process. But still, the 16 sRNA that is a thing which is very small, okay, and they are having a diverse role to play, not only in the phylogenetic studies but also in our body mechanism. So that's why this for having this very specific sequences of this oligonucleotide, we can analyze the phylogenetic relationship between the species. If the species is different, they are not going to have the same kind of oligonucleotide sequences. But if they are different, uh, if they are same, we will be having the same 16 as RNA. But if they are different, we will find the difference. Last thing in the whole taxonomy techniques, this is a thing that is called polyphasic taxonomy. So what happened when we study the polyphasic taxonomy? In this, studying phylogeny based on both genotype and phenotype. So now the more information will we get it, that is the question. So that's why in polyphasic taxonomy, we going to get all the information we can got from the genotype and plus phenotype. Genotype, internal, and phenotype, the external knowledge from any surface. So information ranging when we see the what kind of information we can got from here. Genotype, obviously molecular characteristic. Phenotype, the ecological characteristic. Molecular characteristic, if we are talking about, we are simply talking about here the DNA and RNA. If we are talking about the ecological characteristic, simply we are talking about how the particular species respond to the environment in which they are present. So ecological characteristic and molecular characteristic, they are both important for emitting the ultimate final product of taxonomy, or how we can study the similarity and differences between the two species. This is the whole concept of phylogenetic, and these are the some noble techniques by you can got the whole knowledge of the phylogenetic relationships among the groups of species. These are the references. Even if you guys have any problem in the lecture and you didn't got it clearly by some point, you can go through these references and clear your doubts, and even you can ask me if you have any problem related to the lecture. That's all for today. Thank you.